ASP2138 is a bispecific T-cell engager agent which binds CLDN18.2 and CD3 on lymphocyte. And it activates lymphocyte around claudin-positive tumor cells, and because of the release of cytokine, it results in cancer cell killing. So this agent targets CLDN18.2 because it is already established to target in gastric cancer because of previous phase 3 trial of zolbetuximab, which improved over survival with chemo plus zolbetuximab. Zolbetuximab is a IgG monoclonal antibody which activates NK cell macrophages to induce ADCC, CDC, and ADCP, and different from ASP2138, which activates lymphocyte. So that is an important rationale to develop this agent for CLDN18.2 positive tumors. And in this phase 1 trial, we evaluate single agent safety as well as activity. We especially test both subcutaneous infusion and IV infusion. So first poster which I presented in this ESMO is regarding IV infusion, which enroll both gastric and PDAC population. First, safety profile is within acceptable range. Around 30% of patients experience cytokine release syndrome, CRS. Most of them were with grade 1 and grade 2 and well controlled by established management protocol. Another common event or GI toxicity like nausea and vomiting. It is not easy to conclude, but a little bit lower than that of zolbetuximab in terms of incidence. The overall efficacy is not high in terms of response rate, but 10% patients achieve response, and this was observed regardless of claudin expression, and around 30% additional patients achieved disease control. And interestingly, there is a trend of a higher PK or dose and better disease control rate, which led to us to conclude one dose as an established recommended dose. And then we also tested subcutaneous infusion, which also showed a very similar safety and efficacy profile. Subcutaneous infusion is a little bit less frequency of cytokine release syndrome and better to combine with first-line chemotherapy and the second-line chemotherapy. So we combine it with a first-line FOLFOX plus pembrolizumab and paclitaxel plus ramucirumab. And both cohorts showed acceptable safety profile, although we observed the toxicity which reflect each component of chemotherapy and ASP2138 as well as the pembrolizumab in front line, but overall manageable. Interestingly, more patients associated with gastritis with second-line paclitaxel and ramucirumab, but at this stage we are not sure whether this is class effect, especially combination with paclitaxel or ramucirumab, or just based on relatively small sample size. So we need to carefully follow up with additional patients. And efficacy is very promising with more than 60% response rate with first line and more than 30% in second line, but we need additional follow-up to see additional efficacy results.